Okay, maybe better. So I'm working at a place called ESPCI, just up the hill, not far from here. And so today I will present this talk entitled Tribute to Anthony Genot. So Anthony was a researcher in DNA nanotech and molecular programming. So he had a talk scheduled for this conference, and he very suddenly and very brutally passed away a couple of months ago. And so the organizer kindly proposed to use the slot to present a tribute, and that's what I'm going to try to do. So Anthony was a close collaborator of mine, a former postdoc also, and a dear friend. So I have to admit I'm not very used to do this kind of talk, and I hope I will never have to be used to do it, but I will try to do my best. So I also consulted with some colleagues and friends of Anthony, and together we decided the format of the talk, that the best format for this talk would be to select a few picks of Anthony's scientific path and try to present them to you in the hope that it could be an inspiration for some. So briefly, Anthony was French. He was born in Besançon in the east of France and went to École Polytechnique, which is a famous school in France. And then he did his PhD in UK at the University of Oxford under the supervision of Andrew Saberfield. And then he did a postdoc in Japan. So he moved to Japan to do his postdoc, and then he spent a little bit of time in France, but very soon became a permanent researcher again at the same location in Japan. And so he built a group there, which was very successful, and recently was awarded the bronze medal of CNRS, which is a distinction for CNRS, and became research director in 2024. So Anthony's scientific interests, as I mentioned, were around DNA computing in particular, and he worked on enzymatic and non-enzymatic DNA computing system, which I will review later on. And a bit more recently, he was involved also in the field of DNA data storage, working in particular on the synthesis problem, how to make synthesis faster, but also on many other aspects, different aspects revolving about this idea that one day maybe we will have large-scale DNA databases, and what we would like to do with them and how we would like to handle them, and what new opportunity would come with this new technology. He was also active in microfluidics, in particular using a droplet as microcompartments. DNA nanotech, which is the idea of making nanostructure, encoding nanostructure by the design of DNA, also DNA gels. And Anthony was somebody who had much more ideas, or at least many ideas, than only two hands. So he could not implement, although he was trying very hard to implement as much as his concept as possible. He also just proposed some of them to the community, new theory, new ideas, new concepts that were later on taken and explored by others. So very recently, the most recently was involved in two big projects. One is a molecular archive project that Mark described earlier. And in this PEPR, he was co-leading two work packages, in particular for synthesis and for applications. And even more recently, he was just launching a high-risk and high-reward CNRS project called Calc DNA, about DNA computing, together with Nicolas Chabanel. Okay, so about Anthony's scientific journey. So it starts with his PhD at the University of Oxford in the group of Andrew Tuberfield and getting into DNA computing. So at the time, there was a lot of excitement about a technique to build a chemical reaction network out of DNA using the concept of tow-hole mediated strand displacement. And so tow-hole mediated strand displacement is the thing you see on the right. It was invented by other researchers in 2000. And it is based on the fact that when you have a double-stranded DNA, replacing one of the strands, invading one of the strands by another strand, even if it's identical, is going to be very slow, unless there is what is called the tow-hole, which is here the green part, which is a small part of single-stranded DNA that allows the invading strand to dock and then invade much faster. And so this, in fact, gives you kinetic control over a strand exchange reaction in DNA chemistry. And when you have kinetic control, you can start building a network. So these technologies were refined more and more until maybe 10 years later, after the invention of tow-hole mediated strand displacement, people were able to build these relatively large circuits with computational capabilities. So here I took an example of a circuit that contains a handful of maybe close to 10 gates. And in this case, it can compute, it can extract compute a square root function. So that was more or less state of the art around the time Anthony jumped in. And then, yes, to assemble these networks, of course, you still have to design many different strands, assemble these strands into gates, and put all these gates into a test tube so that each of them can perform its function, propagate the signal, and at the end you get the output. So Anthony, when he jumped in, was not fully happy about the state of the field, and he had some question about how does that scale if really you have to build a different gate with a different sequence made of DNA for every node in your circuit. Is it really going to scale? Are you really going to be able to do large computations? And also questions about energy consumptions, in particular related to the fact that these circuits are used only once. So you build a circuit out of DNA, you send the input, you get the output, but then if you want to do a second computation, you need to build another circuit, and you cannot reuse them. So you had some question about this topic. And also about the architecture that was mostly used at the time to build molecular computers, which is inspired by computer science based on Boolean architecture. So is it really the best architecture to implement computation inside of chemistry? And if it's not, well, what else do you want to do? What do you want to do? And so during his PhD, he started a number of projects. One was about combinatorial displacement of DNA strands. And I just extracted a quote from that very paper. So, and it is related to some mentions that were done in the previous talk as well. If you want to do matrix multiplication, for example, in an n-dimensional system, the scaling grows as n to the cube, because you have to multiply all these different the different inputs in the to compute the full matrix computation, and then the scaling is quite bad. If you have to make a different DNA gate for each of those individual operations, it's going to be very difficult. And so he proposed to use the parallelism of chemistry and to, in fact, just split. Oh, sorry, it doesn't work. So to split the the toehold and the displacing domain in two and put them on different strands, and then make a linking domain so that in fact they could combinatorially reassemble and reproduce all these complex that you need to perform the matrix multiplication, but you have only to synthesize a small number of them. And so you see the experimental implementation on the right side with a small matrix, a 2x2 matrix multiplying a 2 length 2 vector, and the gate reassemble directly computes the result, as you can see down there, and this can be read by fluorescence. So I think it was an interesting concept to use this combinatoriality to limit the number of individual gates that you need in a particular circuit. Another idea he had was to increase the functionality of the toehold mechanism itself by just separating and putting the toehold and the displacing domain further apart, and there is a linker between the two. And then he discovered that depending on the properties of the linker, the kinetic of the reaction could vary a lot. And in fact, it happens that you can change the physical properties of this linker, for example, by switching from double-stranded to single-stranded, or the reverse, which is what you see again on the right side. So here you have a reaction that is very fast when the linking domain are single-stranded, but as soon as you make one of the two linking domain at least double-stranded, you have some constraints that that's going to very slow down very much the reaction, and you have these three curves on the top that are very slow. So now the toehold itself becomes controllable, and you can even think about the toehold of a toehold, which is the one on the bottom, where you can displace and restore the single-stranded domain by using a toehold to displace the double-stranding part. Okay. And then concerning energy consumption, Anthony also thought about making this circuit just reversible and switching from enthalpically driven circuits to entropically driven circuits. So he proposed an architecture where indeed that allows you to create DNA circuits that would dynamically respond to change in their input. And this is what is represented again on the right. You see that it's a single tube, and you provide different types of input. The circuit computes something, goes to the on state or the off state, and you can dynamically change the input, remove one of them, add another one, and the circuit will readapt to those various inputs. So here's the dream of being able to having a real computer to which you can provide various inputs and get various answers was becoming a reality on quite a small system at the time, but I think this idea has been taken over by many, by some at least, to make now larger circuits. Okay, so that was a PhD state, and by the time he finished his PhD, Anthony was, I guess, definitely addicted to DNA, and he moved to Japan and joined my group there. So in Japan, he started to add another component to his mixtures, which was enzyme, and worked with DNA and enzyme-based molecular programming in the framework that we now call the Penn toolbox. So the inspiration there is less Boolean and more biological, and it comes mostly from gene regulatory network. So you probably know about gene regulatory networks, but to describe them in a nutshell, it is this idea that genes control other genes. So genes are nodes, and they're going to control the activity of other genes by sending them regulatory protein, right? So a gene encodes a regulatory protein whose function is just to activate or repress another gene, which itself encodes another regulatory protein, and that makes this kind of large-scale network that biologists study, in systems biology in particular. So the idea was to try to reproduce this concept. So nodes send activators or inhibitors to other nodes to let them express themselves, and that would be the way we control the kinetic. But of course, gene regulation is based on protein expression. It's complex chemistry. You have to make an RNA first and then translate the protein. So was it possible to keep the concept but to simplify the chemistry as much as possible? And so the answer is yes, it's possible. Maybe we can just use simple DNA replication chemistry using a polymerase, for example. And in the end, that became this concept of just 3 enzymes to encode a few basic modules, of which the most important is the first one in blue, which is called activation, and that Nicolas already described, where, so now this blue and red arrow down there would be the gene with its regulatory part in the blue and its coding part in the red. And so if the regulatory part is activated by a complementary activatory blue DNA, it will come, bind, polymerase and nickase, polymerase extension, nickase cut, and the coding part of the gene, which is just an oligo in that case, would be expressed and can go and activate another node in the circuit. So to make it just a bit more precise, I have some slides here. So you see what I've just mentioned. The input comes and activates the node so that it produces the output. That's the most simple, but in some cases the output can be the same thing as the input. So now the input comes and activates a node that produces the input itself. So from one molecule you have 2, and then you get exponential amplification in that case, isothermal exponential amplification. And you can also modify the system so that you produce inactive version of the molecule, and this will be a threshold. It's going to be very important for some functions. And of course, you have reporters too, so that you can monitor these reactions by fluorescent means. So using these ingredients, one of the most simple circuits that you can build, but also a very interesting one, is this one. So you have a node, which is called alpha here, with self-activation. And it can take an input. Input is going to be converted to activate the node. The node is self-activatory. There is a threshold, and there is a reporter. Right, so you have in fact those 4 modules that I've just described chained to one another. And what you obtain in terms of dynamical behavior is represented here. So depending on the amount of the strength of the input, so the amount of this blue input that we have here, which is in that case a microRNA, but it could be any piece of DNA or RNA. So depending on this concentration, you see that the system is going to switch state, but after a given amount of time, and the stronger the activation, the faster this switch is going to happen. And so if you cut at one particular time point, and you look at the state of your system, you get this input-output response function, very sharp switch, which is of course reminiscent of the behavior of a formal neuron. Right? So here we have a neuron with a single input, and it applies a nonlinear function on that input to decide if it wants to go on or if it wants to stay off. So these neurons, as I mentioned, can be connected to each other in recurrent or feed-forward architecture. So we'll start with recurrent architectures here. So here you have two of these self-activable neurons, and they are connected to each other, and they have a tendency to kill, to inhibit the other neuron. And if you think about the behavior of this type of system, you can find out that in principle it can exist in 4 states. Either none of the neuron is active, the 2 neurons could be active, or maybe only one of the neurons is active and it kills the other one. But of course, the fact that the system can explore these different states depends on the parameter landscape in which it lives. And this parameter landscape is defined by the concentration of the oligos, DNA oligos, that encode this particular circuit architecture. So the question when Anatoly came in was is it possible to map this parameter space, and which concentration of one of those particular compounds, how much of one of those particular compounds should we put in the tube so that we get the desired behaviors, so maybe to get, for example, the system in a just bistable state, so it can be only on the alpha and 1, 0 or 0, 1, which is a very classical and useful behavior. But to map that parameter landscape, you have multiple dimensions. You can change different concentration. And if you want to get a good resolution in this relatively high dimensional space, you need to do a lot of experiments. So at that time, Anthony developed a system to use microfluidic droplets to scan a parametric space of a given network topology. And this would work in this way. So we had a microfluidic system that is going to produce some droplets, as you can see here, by flowing an aqueous mixture across an oil channel. And but the aqueous mixture is not coming from a single inlet, it's coming from multiple inlets. And these inlets have different flow rates. So at different point in time, there will be a little bit more of the blue from the blue inlet and a little bit less from the red, and a particular amount from the green. And these ratios are going to change very fast in time, so that every time you make a small droplet, its composition is going to be different from the one before and from the one after. And so with this, because you've put one particular compound in the red tube and another particular compound in the green tube, you can scan the concentration space, in this case in 2 dimensions. The tube in the middle is used for volume compensation, so that overall the total volume remains the same. And of course, these droplets are made at a very high frequency, like kilohertz. So it's difficult to keep track of each droplet and to know what is its content. And so the idea was to put some fluorescent barcodes, some fluorescent species, which are inert for the system, but can be easily looked at under the fluorescent microscope. So that, and there is a red barcode for the first compound, for the first tube, and a green barcode for the third tube. So that by looking at these fluorescence, you can come back to the composition of that particular droplet for those particular chemicals that you know affect the behavior of your system. And if the video runs, yes, you will see the scanning in process. So these are where the 3 inlets get mixed, and the droplets are formed at the top. You don't see them because they go so fast. So this is the workflow that was developed to be able to scan this parametric space. So as I explained, there is a microfluidic part, and then the droplets are connected. They are layered to make a so-called droplet crystal on a glass slide under a microscope, and you see that they have all different colors because they have all different composition. And this droplet crystal behavior now is observed with other fluorescent dyes. And then what is possible is to reorganize the different droplets according to their barcode fluorescent concentration. And so it means that you also reorganize them according to the concentration of the compound that affect the behavior. And what you expect is to be able to extract with this technique the full bifurcation diagram of that particular system in that particular parametric plane. And so this is an example based on this potentially bistable system. So all those droplets have been reorganized in 2D. The two concentrations that were scanned here is called alpha to alpha and beta to beta. These are the strengths of the two self-activating nodes. And so, yeah, if the node alpha is much stronger than the node beta, you expect the system to be always in alpha. If it's the contrary, you expect the system to be mostly in beta, and this is what you see on these two diagrams. But the experiment trick was to initiate the system in two different initial conditions, either with a lot of pre-activated alpha or pre-activated beta, right? And then, if the system is actually bistable, you should find it in two different states, even after a long time. And this is what you can see here. There is a bistable zone where the state of the system actually depends on the initial state. So this is a proof of bistability, and it means that you can obtain the proper behavior bistability for this particular network topology if your concentrations of these for these two nodes are in this particular subspace. Okay, so this was about like connecting nodes in a recurrent architecture. Another option is, of course, to connect them in feed-forward architecture. And by the time Anthony was working on this, like deep neural networks had become very fashionable. And so the idea was to say, okay, can we also look at molecular programming from a much more deep learning perspective using neural architecture? And maybe one application we could think about would be to use them to classify molecular signatures in diagnostics. So for example, you have from a sample of blood, you can extract many different concentrations, and maybe there are some patterns in this multidimensional concentration space that would correspond to being healthy or having a particular disease. And so, for this purpose, the previous node had to be updated a little bit because we needed to add some new functions, and in particular, be able to connect multiple inputs to one particular neuron, and also adjust the weights of those inputs. So maybe the weight of input x1 must be high, and the weight of input x2 must be low. And additionally, some of these inputs may have a negative weight, which was not embedded so far. So this required a little bit of tinkering, and mostly at this first stage, so the conversion stage, but it was possible. So Anthony designed some tricks so that it would be possible to adapt, to adjust the weight, and also to change their signs, which is represented here. And then also we replaced, because we wanted to be able to do multi-layer neural network, we replaced the direct reporting at the end to creation of another strand that can then be propagated to layer number 2, and so on. And so this is a test like a 96 well plate type experiment of a single neuron. In that case, with 2 positive weights on the left side and 1 positive and 1 negative weight on the right side. So what you expect with 2 positive weights is that the system will get on and respond only if the sum of the 2 inputs is sufficiently high. And in the other case, you expect the same behavior to get on only if the difference between these 2 inputs is across the threshold. So you have a diagonal pattern in one first diagonal or second diagonal. This is what you observe in low resolution. And to get higher resolution, of course, it's better to go back to this droplet scanning system. But now we are not scanning the parametric properties of the network. We are scanning the input concentration of that particular perceptron. And so you see the same figure as the one I showed before, I think, by just again, putting the droplets that are generated here back in their position in the network, and we get this diagonal. And because this technique is fast and efficient, it's possible to test different weights, more rates, so we can adjust and give a strong weight to x1, and this will shift, change the slope of the separation, and or use a negative weight, as you can see on the right. And so what was also interesting by this high resolution experiment is to notice that the separation between yes and no response of the system is extremely sharp. And this, I don't know, maybe some of you know a bit about the literature in this field, but typically this is quite different to obtain in molecular system. And the separation is usually relatively shallow and gradual. So here we had a very, very sharp separation. And to show the interest of this, we designed this majority voting algorithm. So it's still a single neural network, but you have now 10 inputs, and you ask the system to make the difference. So each input is just binary, so it's on or off, 0 or 1. But you ask the system to count the number of input, and if majority is achieved, switch. If it's not achieved, you want to stay off. And for this, of course, you need a very sharp separation because you need to make a difference, to change the response according to a difference which is just a few % of total concentration. And so this is what was represented here. So you can think of the input as animal voting, and we could. So these are different patterns that you have represented in each column. Each pattern has a different total number of yes votes, and then exactly at the limit of 5, you see that the system behavior is indeed switching as expected. So then it was time to try something completely different and new, because nobody has ever been into multi-layer neural architecture at the molecular level. So here it's a relatively simple system with just one hidden layer, and this one is going to compute a kind of a gate function, because the first node of the first hidden layer will activate at some particular concentration. The second node of the hidden layer is going to activate at a second concentration. But because the second node, as you see, has a negative output on the final neuron, then the system is going to go back, and what you will see is this type of response. What you expect, at least. And so this, and then you can change in the width in which the system is expected to respond by adjusting some of the concentration for these nodes. And so this is a droplet scanning verification of this behavior. And we look, in fact, at the state of the different nodes. Here, the node alpha of the hidden layer, node beta of the hidden layer, and final node. And see, alpha doesn't, because we are adjusting the response of neuron beta, so alpha doesn't change. Activation of neuron beta indeed switches as we change the strength of this the repression of the bias, basically, of that neuron, and this translates to the response, the positive response of the final neuron. Another multi-layer architecture was also used to do a nonlinear partition of 2D concentration space, which is only achievable with multi-layer neurons. It's not something that you cannot do with single layers. And here you see that one neuron computes a particular partition in green, another neuron computes a particular partition, and then these two everything is combined by the final neuron of the final layer. Okay, that you can see here. Okay, so that's more or less what I wanted to present in terms of the scientific outcomes of Anthony. There is, of course, many more that I have not discussed. There was many other projects. I want to also emphasize that Anthony was himself a node in a network, a scientific network, in between some of his more senior colleagues and mentors, and the group he has assembled over the years, and the students he has taught. Anthony was not just a mind. He was also a very keen experimentalist. And you see on this slide Anthony shopping for consumable and making experiments with those consumables. He was also very much involved in international cooperation and collaborations, in particular between France and Japan because of its geographic location, and always ready to have fun with his friends and colleagues. Before I finish, I just want to send some thoughts to his family, his colleagues, his friends, students. I guess many will miss him, and I thank you for your attention.